Okay, hi. Thanks to the organizers for the invitation to speak here. I've had a great time listening to many diverse talks over the last few days. Because of the theme of the meeting, I'm going to talk about some older work. And hopefully towards the end, I'll talk about something that we're engaged in right now. Okay, so what I've studied, what interests us are these complex behaviors that we call motor skills. And skills can be diverse. They can range from serving a tennis ball, playing a sitar, or just simply tying your shoelace. But what unites these very diverse behaviors is that they're elementally composed of a series of simple movements that are sequenced together in precise temporal order and executed in a very fluid manner. Gaps in between the individual sub movements in order to give you a stereotype motor sequence. And once they've been learned, these skills can be very, very stable and stereotyped. Here's an example of a tennis player who's serving a tennis ball. And you can see over multiple repetitions of a serve, so I've traced her hand in these videos. You can see that the trajectories that her hand follows overlap almost exactly on top of each other, right? And if you now look, so this is actually a video I found from a tournament in 2013. And I have a video, actually, I found a video on YouTube of her coming back to the same tournament in 2014. And you can now trace her hand in a year later, and it basically lies exactly on top of where she was before. In fact, the slight change that you see here in the vertical component is really just because of the placement of the camera slightly different from one year to the next, right? So once these skills have been learned, they can be very reliably executed. And this contrasts tremendously with the behavior of beginners, right? So here's a video of a beginner tennis player who's serving, attempting a serve, I would say. And besides the obvious footfall that this person is doing, you can see that there's a lot of variability from one rendition to the next. Right? So you could sort of define skill learning as the process that takes variable sequence of movements like this and makes them stereotyped. And this takes time, and this results in improvement in performance at a reduction in variability. Okay? So how is this process supposed to happen in the brain? This is what, how does it take place in the brain? What are the circuit changes that underlie this? This is what we're interested in answering. Okay, and so there's a standard model of skill learning, or rather of motor control more generally, in that the brain is, motor circuits are hierarchically organized, that there is a circuit somewhere in the brain that makes a decision about what action to pick, and this is based on what the state of the body is, what is the sensory input it receives, what kind of reward information has been tied to these various actions, and then sends that command to a motor control or a series of motor control circuits that translate that decision into the spatiotemporal patterns of muscle activity that are actually required to execute that movement. Right? Now, early in learning, it's thought that the sequence of movements is driven by dynamics in the selection circuit. Right? So you're sort of selecting one movement and another movement, et cetera, et cetera. And this is quite a variable process. But over time, the sequence of movements gets consolidated within the controller, so that the selector is now selecting a sequence of movements rather than an individual movement. And this is why you can do this much more precisely and autonomously. Okay? And also, early in learning, you might notice that you have to really think about what you're doing. But late, when you've practiced a behavior over many years, you can just start it and it happens by itself, right? So you don't really need cognitive control. And this is thought to reflect that process. And if you actually try and map this hierarchy to the brain, and this is just a cartoon of the motor system of the brain, it's thought that this selection circuit, I mean, there are many selection circuits, but in the context of this form of learning, it's thought that the basal ganglia are where the selection process happens. And the motor cortex, and especially motor cortex, is where the improvements in the skill are stored, right? So that you can execute this more autonomously. However, this model has come into a lot of questions, primarily because actually of a lot of work in songbirds, where the basal ganglia seems to play a very different role from what it's hypothesized to play in the mammalian brain. So and you've seen a lot of talks on the birdsong, on the songbird brain, so I won't go into too many details, but the songbird brain has a motor pathway and then also an anterior forebrain pathway, and which involves the basal ganglia, this region called area X, that's a region of the bird basal ganglia. And this pathway is actually important for learning the song, but not really for producing it once it's been learned. And one interesting series of experiments have shown that this anterior forebrain pathway can actually generate a bias in the motor output, right? And this is actually dependent on area X also. And this bias is actually more consistent with a role in control, at least temporarily, before it gets consolidated into the motor pathway, right? So, in other words, the basal ganglia can do more than just select actions. And we want to understand really what happens in the mammalian brain when the skill is being learned, and what is the role of the BG. Okay? So first, because most rodent studies typically just associate lever presses with reward, or simple associations of actions and rewards. We really want to train skilled movements in rodents, similar to the kinds of skills you can train in humans or you can train in birds. Right? So what kind of behavior can we pick for that? So here's the task that actually trains a skilled behavior, and you'll see why it's a skilled behavior in some time. So the task is very simple. A rat has to press a lever twice with a prescribed time interval. So only if it presses at twice with the interpress interval of 700 milliseconds does it get reward. Otherwise, it doesn't get reward. And so early on, what happens is that rats, so this is a density plot of the interpress interval as a function of the training history. So this is early in learning. And so you can see that rats tend to press at much faster intervals because their natural behavior is just to bang away at the lever, right? And so what we do is we don't want them to get no reward in the beginning. So we have a pretty generous range of interpress intervals that can be rewarded. And we slowly shape the behavior so that we keep constricting the range of intervals that they can get rewarded on. And eventually they learn that 700 milliseconds is the optimal interval for the task. And this takes actually about a month. So it's quite a slow process, similar to skill learning in humans, right? So what's actually remarkably interesting about this behavior is that they don't solve this task by just pressing the lever once, waiting 700 milliseconds, pressing again. They actually solve this in the motor sequence domain. And by that I mean, I think it's best to just show you what I mean. So here are 4 independent trials of the same behavior. The rat has just received reward for its previous trial. It's going to go up and engage with the lever. This is the lever. And every time it presses the lever, you'll see a white square appear on the screen. Okay? And it'll play in real time first. It's actually too fast to really resolve. I'll slow it down so you can appreciate the details later. Okay, so I'll explain in the slowed down version. So here's the first lever press. Right? And it's doing a series of very complex movements in between the first and the second lever press. So we don't impose that at all in terms of the experimental conditions, but it turns out that rats pretty much always do something complicated in between. They very rarely press the lever, wait, and press again. No. So they just do this. It's an idiosyncratic behavior. So each animal has its own particular sequence of movements. They're obviously superstitious. They have nothing to do with the reward we're giving them. So here, this is one rat. Here's another rat that has a very different motor sequence. Again, this is in real time. And this is now, I'll show you a slowed down version. So here's the first lever press. And then it actually licks once. And then it does this DJ action, and then it presses again, right? So basically, yeah. 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 No, so this is after learning. So early in learning, it's very variable from one. Yeah. So the variability that you see here, early in learning, is very similar to the variability in human behavior. Yeah. Yeah. Essentially, the reason why this happens is because rats don't really know it's a timing task, right? They just do something that works, and what works is a sequence of movements that happens to tile the interval between the first and lever press exactly, right? And those are the ones that get selected in the reinforcement-driven process. Okay, so having trained animals on a complex motor sequence task, much like say songbirds or human motor skills, we can now sort of test this classical model of, are the basal ganglia selecting movements, and is the learning actually taking place in the motor cortex, right? And so what we did was to actually just lesion the motor cortex in animals that had been trained on the task and ask whether animals could still perform the same motor sequence that they had learned without motor cortex. And so that's what you're going to see in the next video. So on the left are examples of the motor sequence that they perform pre-lesion, and on the right are 2 example trials of the motor sequence post-lesion. Okay? And I think what you'll appreciate this place, is that they're essentially the same. Right? So this was a complete lesion of motor cortex. In fact, you can lesion much of the frontal cortex. And there's actually no deficit at all in terms of the timing, the complexity of these sequences. In fact, they can even learn new sequences after having lesion motor cortex. Right? So clearly, this model is a bit simplistic and perhaps not entirely true, that the motor cortex is not really where these movement kinematics that are important for performing the learned motor sequence are stored. And in fact, there's probably some subcortical circuit where these are stored. And so we sort of decided to, or I decided to rigorously test the role of the basal ganglia in this process. Okay? And because the basal ganglia is also connected to another set of brainstem of motor control circuits in the midbrain and brainstem, okay, which unlike the motor cortex are evolutionary much more ancient. They're in all vertebrates. And so what we did was to record from the basal ganglia. And this is how we record from the basal ganglia. So the animal has an implant of the electrodes targeting the basal ganglia. We're recording from neurons in this area. At the same time, we're tracking the movements that the animals make when they're solving the task using these markerless tracking methods. And so what you'll see are the trajectories that the animal's limbs and head follow, and a raster plot of activity of one example neuron. Okay, and so this neuron tends, and so this is the time in the sequence, right? You can see that there's a stereotype motor sequence that the animals are performing, but there's variability from one trial to the next, right? There's some variability in the timing of these particular elements that compose the sequence. And there's also some variability in the number of spikes and the timing between the spikes, right? So what we can do now is to try and relate the variability in the movement to the variability in the firing patterns and try and infer what is this area actually encoding about the behavior, right? So that's an example of, this is a representation of that particular neuron, right, a raster plot. And you can see that this neuron is actually active at the beginning of the behavior, right? So based on this, it looks quite similar to this idea of the basal ganglia as an action selector, where it triggers a whole sequence of movements in some downstream controller, not motor cortex, but maybe somewhere in the brainstem, right? But actually, if you look at other neurons, they're active throughout the motor sequence, right, at different intervals, at different delays. And so this neuron is active in before the sequence begins. This neuron is active in the middle of the sequence. And so they really form a nice temporal pattern that tiles the entire sequence. And these are some interneurons, right? So this is really not the picture that, in fact, it looks much more like a control circuit would. Right? Now, if this is really reflecting some sort of control process, we would expect to be able to decode the behavior, or we would expect that this activity actually encodes something about the details with which, details of the behavior, right? The kinematics that define the behavior. And so what we can do is, this is just a summary. So this is all the neurons in one animal, and you can see that they tile the sequence quite well, and there's no increase in activity either at the start or end compared to the average. Yeah. So what we can do to test whether these activity patterns encode kinematics is we can take a slice of activity on a single trial, a time point in a single trial, and then ask whether we can reliably decode the ongoing movements at the same time, right? So in this case, it would be the velocity of the hand. Okay, and so this is what the behavioral data looks like. So I've just arranged the velocity of different trials by the length of the sequence. So short trials are on the top, bottom trials at the longer trials at the bottom. The color indicates the velocity, whether it's going up or down. Right? That's basically what this is telling you. And you can see that there's an overall structure to the sequence, right? That there's a particular pattern associated with the sequence. which scales as the sequence lengthens, but there's also a lot of local variability, right, which is not explained by this global scaling of the motor pattern. Now, if we actually try and we can actually decode this very reliably from just looking at these activity patterns, right? So this model doesn't have information about the behavior. I mean, at least not behavior at the previous time point, but we can actually decode the ongoing behavior very reliably. Now, you might think that this is actually quite a so this really this may not necessarily indicate that these neurons are encoding the kinematics of the behavior, right? Because it's a stereotype motor sequence. That is, the animal performs the same movement at the same time within the sequence, right? The same phase of the sequence. So as long as you have a good temporal basis for the behavior, you can decode the kinematics even without the area necessarily encoding the kinematics. And a good example of this is, so perhaps what's happening is that the basal ganglia is just doing action selection on a much finer time scale. Quite similar to how the HVC activity, right, which is sort of provides a clock-like input to organize the birdsong behavior, right? And RA is really where the kinematics are encoded, right? So the question is the basal ganglia activity a lot like HVC, although they are quite different structures? And so to answer this question, what we can take advantage of is the fact that I sort of lied a little bit to you that I sort of gave you the impression that animals learn a particular motor sequence. And that's the only sequence they perform once they've become experts. But in reality, there are slight variations of the sequences, which correspond to slightly different behaviors. And you can actually visualize this. If you take all the kinematics, the movement kinematics, aligned to the two lever presses, and then project them into some low-dimensional space, you can actually see clusters popping up, right? And so if you then do some clustering of these of different trials, you find that sequences these are discrete, there's sort of variations on the sequence, right? Distinct sequences that pop up, in which typically the start and end is the same, but there's some different movement in the middle, okay? So the sequences start and end pretty much the same way, but there's typically some, like for example, here they have an additional movement in the middle, here they lack that, here there's some other movement in the middle, right? And just to give you a concrete example of this, here's two different sequence modes, we call these modes, where you can see that if you align the trajectories from the start to the first lever press, they start and end the same way, but there's a slight difference in the middle. So we can now ask, the sequence, we can ask whether neural activity in the basal ganglia, does it actually encode these kinematic variations between sequences, right? And here's some ensemble activity. So these are different neurons. And what I'm showing you is the average activity pattern of that neuron in each of these sequences. And these symbols, so the square indicates the time at which the sequences start to diverge. The star indicates when they maximally diverge from each other. And you can get a hint that this is actually the same pattern that you see in the basal ganglia as well, right? And actually, perhaps you can see this a little more clearly in this other representation. So these are two videos of the actual, the two sequences that I'm talking about. And you can see the kinematics up here, and this is a low-dimensional representation of the trajectory that the neurons actually follow in this state space, right? And I'll play this a few times in case it's not clear. But you can see that these sequences are very similar, but they have a different element in the middle. And the neural representation also follows the same trajectory at the start and at the end of the sequence, but in the middle, it shows a nice divergence. And in fact, the divergence between the two sequence modes, both kinematically and neurally, really align on top of each other, indicating that this representation is a kinematic representation. Okay. And then finally, what we can do is also just ask, is this area required for performing the motor sequences? Like motor cortex wasn't, right? So we can lesion this area in trained animals. And what we see is, so that's the red curve here, we see an immediate drop in performance after lesions, very unlike what we see in motor cortex. And if you look at the trajectories that the animals, so these are all individual animals in different colors. So pre-lesion, each animal has its own solution to the task. So they were, because they're idiosyncratic, right? Like just like humans learning a tennis serve, each person has a slightly different solution to the problem. In fact, that's why you have coaches so that you get the same consistent solution. But rats don't have coaches, so they have a large variety of solutions to this motor sequence task. But after lesion, they just resort to this very simple lever pressing behavior that you see very early in training, indicating that they've essentially forgotten what they've learned. Okay? And so just to summarize this, I would argue that this tells us that it's not that complex behaviors need not be cortical. There's a pretty rich subset of cortical circuits that can support such behaviors. And secondly, I also think one more takeaway that I have personally from this is that I think we tend to be quite simplistic in ascribing particular functions to particular areas. But a lot of these areas actually talk to each other in terms of loops. Right? So there's a motor cortex, basal ganglia, thalamic loop. There's also a loop between the basal ganglia and brainstem controllers, and then back to thalamus. And then so these loops perhaps implement a lot of these functions. And then the question is, what is each area doing in that loop? And can we actually talk about it having a distinct function from a downstream area or an upstream area? Okay, so that is some of the past work. Are there any questions? Because I'll be switching gears. Yeah.
In the kinematic variables, you chose different kinds of variables, right? At some point, you showed us a sequence which was correlated to the forelimb velocity. And later on, when you're showing the kinematic sequence, you were looking at position. Did you arbitrarily try like all sorts of kinematic variables and found different neurons were encoding different variables? Or did you find that most of them would encode one particular kinematic variable depending on where you're recording from?
Yeah. So in general, for the analysis that I've just shown you examples of particular kinematic variables just for illustration. But in reality, we actually try to decode multiple variables, and the accuracy measures we report are across many variables. And also for the defining the sequence modes, we actually take into account all the variables: position, velocity, acceleration. But regarding the question of what do neurons encode, that's a little hard to answer with this kind of behavior, just because the variables themselves are so correlated, and the behavior is so idiosyncratic that it changes dramatically from one animal to the other one. And so for that, I think you really need a task where they can flexibly recombine different movements in a way that we want rather than what they choose to do. Yeah, exactly. They can be quite variable. So as I showed you, so if one animal used its tongue, they use their hind limbs, forelimbs. The head is also often used. So actually, we have an accelerometer on the head in the implant, and we can get very good readout of the motor sequence just from the head movements also. So it can be quite diverse. So they don't seem to sort of converge on. Of course, they use their hands a lot because the task involves using your hands to press the lever. But in between, you get all kinds of movements. Okay, so I'm going to shift gears a little bit because I have my own lab at IISc now. And the question that interests me a lot is this question of how are these complex motor sequences or complex behaviors in general learned in the first place, right? So one of the challenges in studying skill learning is that it takes a long time, right? It takes many, many renditions or many, many trials for you to perfect a behavior. And that can take weeks, months, and sometimes years. And so the issue is that we actually lack, in systems neuroscience, we lack, first of all, methods to easily train these behaviors, because unlike songbirds, these are actually trained behaviors. We need to motivate animals and train them every day. And secondly, we lack tools to follow the activity of a consistent set of neurons over these timescales. So the way we typically do the experiments is that we put animals in a box, and then they do the task, and we record from some neurons, and then we take the animals out, put them back the next day, and then typically there's a different set of neurons we're recording from on that day. And so we don't really get a nice, consistent picture of what's happening over the learning timescale. And so what we've done in the past, in the recent past, is to develop rodent models of motor skill learning. And these rely on some platforms that we've also had to develop. One is an automated behavioral training setup. So this is essentially an operant conditioning gym. So each rat lives in its own home cage. There's a series of home cages. We have about 80 right now in our training facility. And rats live there and they're trained in a fully automated manner. And we can do this on a high throughput scale. Right? So these are all operant conditioning boxes. They're all controlled by Raspberry Pis and microcontrollers. So the training algorithms implemented in these things. And the training is fully automated. So we don't have to actually, as experimenters, we don't have to deal with changing the task conditions from one day to the other. And so this really allows us to do high throughput neuroscience. The other thing we had to develop was a way to record from the brain over long time scales. And so we took one of these automated training cages and outfitted with the electrophysiological system where the animal is continuously tethered, right? And so we can record from the brain 24/7 for many months at a time. And now this is not routinely done because what happens is you have to process this electrophysiological data in some way. So these are some high-pass filtered recordings from these electrodes in the brain. And so you can see these action potentials very clearly on the different channels, right? And the typical way to process such recordings is that you apply some sort of threshold to isolate the action potentials. You then, then you project those action potential waveforms that you've captured into some feature space, right? Say the height of the action potential on electrode 1 versus electrode 2. And then you see these nice clusters popping up, and each of those clusters corresponds to spikes fired by a single unit. Right? So you can apply your favorite clustering algorithm and recover the action potentials or the activity of individual neurons from this mass of multiple neurons. But what happens over time is that the spike waveforms change, and this leads to a drift in the clusters over time. And so this is a very challenging problem. Yeah, so this is a challenging problem because you need to be able to cluster a non-stationary data set over time. But we have managed to solve it to some extent. So we have an automated clustering method that can automatically identify neurons despite changes in their spike waveform and track them over days and weeks. So I'll just skip the rest of this so there's no. And so what are the questions we're applying these methods to solve? So the questions that really interest me is how does the brain learn in large action spaces that are characteristic of complex tasks? So a tennis player, for example, has to pick the kind of serve to perform, or has to learn which serve to pick at a particular time in the match, but also how to perform the serve. And that involves learning to adapt or adapting multiple kinematic variables, right, which are continuous. And so this is a very high-dimensional continuous search problem. And what's remarkable is that the brain can do this using very simple feedback. So often the only feedback you really get in most tasks that is useful is whether you did something correctly or not, right? So the feedback is binary or graded in a manner. And so the question is, how does the brain do this? And the typical framework within which we ask this question is that of reinforcement learning theory, which is a computer science field, which essentially asks, how can an agent take actions, or how should it pick actions so that it can improve reward that it receives, right? And typically this question, that is a neural basis of reinforcement learning, is studied using very simple tasks, where animals just choose between two choices, right? And these are called bandit tasks or multi-arm bandit tasks, but typically they're just two-arm bandit tasks. And so what we want to understand is how does the brain solve these types of tasks, but of much greater complexity? And this is a relevant question because there's a lot of machine learning tools now, or there's a lot of advances in artificial intelligence that have designed reinforcement learning agents that can actually now meet or even surpass the capacity of humans to solve particular tasks. But they learn in a very different way. So for example, these are some dated examples now, but I think the same rules apply. The kind of experience this particular agent needs to learn how to handle this cube, right, is worth about 100 years, and that's really not much longer than a typical human lifetime. Similarly, these agents trained to play chess or Go, they've had to play millions of games to really reach human level performance. But in contrast, let's say with a grandmaster, there's an estimate that you need about 10,000 games, chess games, to reach that level of proficiency. Okay, so we're much more efficient at learning with limited data or limited sampling. And the question is, what are these algorithms that the brain uses to learn or to solve such tasks? And so what we do is we've sort of introduced different kinds of bandit tasks where rats say have to choose between multiple options, not just 2, but 4 or 6 or 8. And those options can have, each of them has a different reward probability, and they can be structured in some way. For example, you can have a spatial structure, or they can be unstructured. And then we can ask how animals solve these tasks. We can have continuous bandits, where the action space is continuous. So here, an animal has to press a joystick at different angles, which is a continuous kinematic dimension. And then we can have the reward zone that they have to find by trial and error, jump around. And animals are pretty good at solving these tasks. This is some examples of a rat solving this multi-choice task. And we keep the environment very dynamic, so they have to keep learning all the time. Similarly, this is the continuous bandit, where animals have to alter their motor patterns, their movement kinematics to solve this task. And the nice thing is with automated training, we can get huge data sets from individual animals, up to 500,000, 600,000 trials. And this allows us to do really detailed statistical modeling of this behavioral data to uncover strategies that they use to solve such tasks. So for example, we can sort of fit very large regression models to the behavior. And typically reinforcement learning is very hard to infer strategies from because it's very noisy, because variability is very inherent to the process of learning. And so it's very hard to kind of see signal amongst this noise. But using these large data sets, we can fit models of this sort, and these are interpretable models. So this is just a regression model with a bunch of interaction terms between action and reward. And then we can use this to basically infer the kinds of trial-by-trial algorithms that they're using to solve such tasks. For example, this is one thing we found that rats use in these continuous action spaces, an actor-critic algorithm, where they essentially they use a pretty simple policy, which has a mean and some standard deviation, some variance, and they sample action space with this policy, and then so the reward. So there's a mean angle, there's exploratory variability. What we find is, and this is too much detail for this talk, but they actually regulate the amount of variability. So they explore in a dynamic manner. When they don't get reward, they increase exploration. When they get reward, they decrease exploration, right? And so we can infer things like this just from data alone. And then we can look inside the brain using tool systems neuroscience and ask how these strategies are actually implemented in brain circuits. Okay, so that's just a way to close the talk. So much of the work I showed you was done before, but some of the people who worked on these newer tasks are highlighted here. Yeah.
You look at the reinforcement models, are the rewards stochastically given, or do they get a reward each time? And does that make a difference to the policy that you?
Yeah, so it likely makes a difference. In this case, it's in the decision-making task. Here, it's explicitly probabilistic, and what varies across the different reward choice sports is the probability of reward. But in this task, it's technically deterministic. So if it gets an angle within this reward zone, it should get 100% reward. But because there's motor noise, effective will become probabilistic.
I guess what I'm also asking is that do they always get reward? So there's a probability in terms of the distribution of how often they will get the reward. But do they always get reward each time as long as this probability is maintained? Are they have you introduced stochasticity in terms of just noise of whether or not they will get reward at all?
So that is inherent in this task because it's probabilistic, but we actually do have some trials in which we probabilistically reward them, and that we do that explicitly because we want to break the correlation that typically exists between the action and reward.
And does that affect the policy at all?
So we do it very infrequently. And so it doesn't really affect the policy. But in general, if you give probabilistic reward, so these are called Bernoulli bandits. they're much harder tasks to solve. And so previously people have done tasks like this with humans where they actually varied the amount of reward. But from a single trial, you could infer how much reward you get at one location versus another location. So the exploration wasn't really challenging in that case. But this is more typical of more real-world tasks where exploration is challenging, and even learning the value of a particular pod requires multiple samples from that pod. And so we see very different behavior in such tasks from what people have seen previously. And so we've done similar tasks with humans as well, actually, and even human behavior differs between these tasks and previous tasks.
They've done this with bees as well, multi-banded problems with honeybees.
In the previous part, when you had the complex task and you did the PCA to decompose the motor patterns, I was wondering if you could do the decomposition with simpler elements that could be used by different individuals and use a common simple base. Would that be possible?
I think given a large enough data set, it would be possible. And definitely we have large data sets from individual animals as well. So actually the kind of methods you have pioneered would be very useful to apply to a data set like this. We had tried applying, so Robert Datta at Harvard Medical School has this, his lab has come up with some sort of autoregressive HMM technique that tries to infer these syllables. So they use it for trying to discover sort of syllables or gestures, I suppose, in recordings of just mice, video recordings of mice just running around in a bucket. And so they claim to infer things like left turns, right turns, upward movements. So we tried doing that, but it wasn't very successful because I didn't think it didn't have a biomechanical model in there. I think having some sort of biomechanical model or some constraints of that sort would be useful. Something with physical realism as opposed to just some data-driven thing. Yeah. So I skipped that part. The motor cortex, all this are required for executing the task once it has been learned. And that's because the task doesn't involve dexterity, doesn't involve fine motor control. It is required for learning the task. So if you lesion the motor cortex in naive animals and then train them on the task, they never learn the task. So there is a role for motor cortex in learning. And in fact, if you record from the motor cortex, you see very nice time-locked activity patterns, which you can also use to decode the kinematics of the behavior. But so even though these activity patterns are present, they're not required for the task. And so I think this tells you that, the lesson for me is that you should always do some sort of causal experiment and get rid of the structure in some way to really test whether it's required or not. Because these days people have been finding, reflections of movements in all kinds of circuits, including visual areas, at least in rodents. So just because you see a representation of behavior doesn't mean that area is actually required for that behavior.
The comment is in the songbird, people have used temperature, changing temperature to understand how dynamics are, or where the dynamics are.
Yeah.
And I wonder whether that would be helpful in here also.
Yeah, so actually the lab I did my postdoc in, they were trying to use some of these Peltier cooling devices inspired by the songbird work. But they were not able to just selectively cool the striatum without affecting other areas, which is I find it surprising because songbird brains are much smaller and yet you're able to cool just HBC and not, RA and other areas. Maybe because it's very distinct in its spatial location.
Yeah, there is some increase in temperature in the rest of the brain. But I think a lot of the interpretation was based on how much is
Okay. Yeah. So the striatum is a very large structure in the rat relative to the brain. So you need a very large probe to cool all of it. And then you'll end up cooling most of the brain. So that's the problem. Okay, thank you.